ladies and gentlemen, distinguished lecturers, and welcome back to another set of very interesting talks. And I think we all feel really privileged to have these fantastic speakers, of course, and in all, but also all the speakers that are invited today. It's really a privilege to hear all this news from the forefront of science. Next speaker is Professor Don Canfield, who is a professor in ecology at the University of Southern Denmark. And he is very well known on his work on ocean chemistry, and is actually a person who also got an ocean or an ocean state named after him by Professor Andy Nall. And that's the Canfield Ocean. So please.
We'll talk about that. Well, thank you very much for the kind introduction. It's really an honor and a privilege to be here to celebrate Andy's great prize and award. And I guess on a personal note, we were just talking about this, Andy and I. I've known Andy for a long time, about 30 years. So I was a young postdoc when we first met. And I guess Andy must have seen something in me at that time, because he's been one of those that has actively promoted me and championed me through the years. And I thank you very much for that, Andy. And I get these niceties out of the way, because you're going to be completely annoyed with me by the end of the talk, I promise you. And the one last thing about Andy, because nobody's mentioned it so far, is that you've probably noticed this, but somehow Andy manages to look like a teenager, no matter what. Okay, so thanks, Susan, for your talk, because much of what I'm going to say is going to springboard very nicely from what you had to say. We'll end up in a slightly different spot, but I think that's okay. That's just all good for discussion. So we're going to talk about the evolution of eukaryotic ecosystems, as Susan also did, but from a slightly different perspective. And to just retrace a little bit where Susan has gone, we need to sort of set the stage for what I would like to talk about. And to do that, we need to think a little bit about the evolution of eukaryotes. Susan went through this, and I'm going to go through it just again with a slightly different spin. There are a couple of different models for how eukaryotes evolved. The first one here we're not going to talk about because it doesn't really matter, just for completeness. This is the model that Susan presented to us, where we have eukaryotes springing out from a group of archaea called the Asgard archaea. These archaea have some special membrane properties which at some point allowed them to phagocytose, engulfing an alpha proteobacteria, which became the mitochondria. And from there we have the LECA, the last eukaryotic common ancestor, springing out into the various eukaryotic groups which we're familiar with. Now, as Andy also pointed out in his talk, there are a variety of different ways that eukaryotes can live, but the three that we really want to think about is osmotrophy, which is the uptake of dissolved organic matter. There is phagotrophy, which is the uptake of particular organic matter, and then there's phototrophy, photosynthesis. And based on how we imagine this transition to have happened, if this represents at least part of the stem group of eukaryote evolution, then before the incorporation of either the mitochondria or later the cyanobacteria that became the chloroplast, we're talking about organisms that must have lived, if they're eukaryotes, by osmotrophy, by the uptake of dissolved organic matter. So this also was presented by Susan, and she went through it very nicely, so I don't need to present very much of this. But one can make predictions about what organisms were doing based on their relationship to LECA and the incorporation of the alpha proteobacteria, which became the mitochondria. In other words, the introduction of phagocytosis as a eukaryotic trait. So in this model here, I think it would be safe to say that through this part of geologic time, the eukaryotes that were in the environment should have been osmotrophs, because they hadn't developed the ability to phagocytose yet. And in this model, all possibilities are there. So in that context, what I want to do is to pose a couple of very basic questions. And these questions are, when did the eukaryotic traits of phagotrophy and phototrophy become prominent ecosystem functions, recognizing osmotrophy as sort of the primitive, most available eating style before these? And then, importantly, and something that Susan talked about quite a bit, how important were eukaryotes in carbon cycling during the Earth's Middle Ages? So these are the two things that I want to address. And you guys have seen this already today. Much of what we know and understand about the evolution of eukaryotic ecosystems through time is based on this work by Joachim Brax, where he very carefully and systematically looked at the record of biomarkers as extracted as bitumen from rocks of different geologic age. And there's something that hasn't been mentioned yet here, and that is that one thing you need to know about Joachim is that he is actually famous for not finding steranes where other people have. And what that means is that his work is meticulous. He is incredibly careful in how he approaches his analyses, and he's careful about the way that he deals with contamination. And his records have become sort of the golden spike in terms of how we view eukaryotic ecosystems. And as Susan and Andy have both pointed out, this record is very clear that when we're talking about Ediacaran times and younger, the sterane record is consistent with the presence of a complex eukaryotic, at least phototrophic community with red and green algae in it. When we go into the Tonian record, there has been the identification actually of at least, if I remember right, red algae here. But if we go in the Tonian record, this algal component is basically vacant from the extractable bitumen record. So this is sort of, in many ways, painted our interpretations or at least painted the pathway or the way that we interpret the evolution of eukaryotic ecosystems through time. Now, this is something that Susan also pointed out. Another way of looking, of course, at the evolution of eukaryotes and eukaryotic ecosystems is to look at the microfossils that are preserved. And I don't need to go into this in any great detail, but the point is that there are well-preserved microfossils which are identified as eukaryotes back to about 1.6 billion years ago. So eukaryotes were a part of the ecosystem as far back as then. And this is true also with other types of eukaryotes which can be identified. And if we go to an ecosystem at 1.4 million years ago, this is from the Jameling Formation. There's a variety of different forms which have been identified as eukaryote. The point being that eukaryotes were part of this ancient ecosystem. The other point which I would like to make is that these organisms, many of them tend to be large. And that's going to be the entrance point really into our discussion. It's something that Andy pointed out in his presentation, but I would like to take it just a little bit further. And Shuhei and his colleagues have actually compiled, I think, a very useful data set at looking at the size of, these are acritarchs through geologic time, and at least through much of geologic time. And I must say that this, and Shuhei would say this too, this is not a systematic exploration of the size of acritarchs through geologic time. This is an exploration of what the published photographs of acritarchs through geologic time provides. So it's not necessarily indicative of how the size spectrum of acritarchs have evolved through time, but it may represent it. That systematic study hasn't been done. But anyway, if you can see the scale here, we're talking about sizes in the year zero. These are 20 micrometer bins here. We're up to 400 micrometers at the large end. And you can see that many of the organisms that we're dealing with, many of the acritarchs that we're finding in the geologic record through the late Paleoproterozoic, through the Mesoproterozoic, Neoproterozoic, are actually quite big. And and actually, there's something we could maybe talk about later. There seems to be an interesting trend towards smaller acritarchs when we go into the Cambrian. That could be quite interesting. I'm not going to dwell on it, but if people would like to discuss it, I think that might be fun. Of course, there's a rabbit hole here, and a rabbit hole that I think we have to sort of skirt around just a little bit. The implication when I show something like this and I start to talk about size is that eukaryotes have a particular size spectrum, because what I want to try to do is distinguish somehow in the modeling that I'm going to be talking about between eukaryotes and prokaryotes, at least in a general sense. So the prejudice would be that eukaryotes are big and prokaryotes are small. And if we go to modern active pelagic ecosystems, that's basically what we find. This is a size distribution of prokaryotes from the Ross Sea, and this is the sort of the median here and the size, and this is 1 micron bin here. So basically, prokaryotes, pelagic prokaryotes, tend to be 1 micron or smaller. They tend to be small. You see that here in the Ross Sea. You see that in the Mediterranean, where the micron range would lay about here. So prokaryotes tend to be small, but that doesn't mean that there aren't prokaryotes that are found in the modern environment. And here we're dealing with the very well-known Thiomargarita namibiensis. Here we have some purple sulfur bacteria. Here we have some Thioploca filaments. Here we have a cyanobacteria. So we do actually find large prokaryotes. So there is an exact cutoff between prokaryote and eukaryotic size. One thing I'd like to mention, though, for those that are interested in this sort of thing, we might be able to, in the geologic record, be able to differentiate or identify large prokaryotes by ways that we might not normally think about. Of course, we can identify filaments in the geologic record, and if we exclude filaments, then we take filamentous cyanobacteria and things like Thioploca out of the picture. But these organisms here, like the purple sulfur bacteria and the Thiomargarita, they're living with under specific environmental circumstances by which their large size actually gives meaning. It makes sense. And if we can identify the ecology or the chemical environment where the organisms are living, we might be able to argue whether or not these organisms were present. This is an aside. The point being, though, despite the fact that we do see large prokaryotes, I think it's fair to say that this record here is showing us a lot of large organisms, which certainly must be eukaryotic in nature, and many of them have been identified as that. So why am I going on and on about size? Well, I'm going on and on about size. Actually, Andy mentioned this in his talk, because the lifestyle of an organism, in many ways, eukaryotic organisms, but also prokaryotes, is determined to many ways by its size. So, for example, organisms that are osmotrophs, that means organisms, whether they're prokaryotes or eukaryotes, that are extracting nutrients as dissolved organic carbon, are small in size, and this has to do with the nature of the surface area of the organism, the transporters involved in transporting the dissolved organic matter into their bodies. Now, as organisms get a little bit larger, the photosynthetic apparatus becomes more efficient at accumulating carbon than osmotrophy. So when you get to larger size organisms, the organisms that are actually able to get carbon by photosynthesis become better competitors in the environment for the available nutrients. As we get to larger organisms, nutrients start to become limited, and then organisms that are able to phagocytose, able to take particulate organic matter into their cells, into their bodies, become better able to compete for carbon and resources. And then heterotrophs, those organisms that are living in a eukaryotic sense by phagocytosis, if we're talking about single-celled heterotrophs, tend to associate with the larger organisms. So there's definitely a size relationship between the type of organisms that live in the environment. And again, coming back to a point that Andy made in his talk, is that energy also matters. So this is a plot showing the proportion of total biomass in the picoplankton. That would sort of be viewed as the prokaryotic end of the spectrum, as a function of nutrient concentration, in this case nitrogen. And you can see that as the nutrient levels increase, the proportion of total plankton in the picoplankton range decreases dramatically. In other words, you get an evolution to larger organisms in larger size classes. So size sort of is an indicator of the types of lifestyles that organisms have, and the amount of energy impacts the size spectrum. So what I'm going to do is I'm going to use sort of modern developing models for size and trait-based ecosystem structure to analyze this problem from an evolutionary perspective. And these are models that are used by Mick Fallows and others that are trying to understand the dynamics of modern ecosystems. And one of the interesting things about these models is that they don't look at ecosystem structure from a species-specific perspective like a lot of the older models do. What they do is they look at traits. And these traits are whether an organism lives by osmotrophy, phototrophy, or grazing. Those are the traits. And what happens is that there are a whole bunch of size-specific allometric relationships that can be extracted from how modern organisms work, coupled with some aspects of physics and diffusion and stuff like that, which can be used to develop what you would call very generic models about how nutrient and energy flow will impact the structure of a marine ecosystem. So basically, we have carbon gains by osmotrophy, DOC uptake, phototrophy, grazing. We have carbon losses by virolysis, respiration, and grazing. Any organisms can possess any or all of the traits. In our particular case, we're modeling these ecosystems as in a chemostat kind of a way. And in doing that, what we need to do is we need to calibrate the chemostat to parameters like nutrient levels and mixing rates that we find in the oceans. I'm not going to go into that into a whole lot of detail now, but that's just how the model works. And what's I think kind of interesting about this model is we're actually able to test specific evolutionary scenarios with it. So scenario number one that I would like to test is we have a picoplankton phototrophic community. In other words, we have a phototrophic community which is dominated extensively by cyanobacteria. We don't allow grazing, but we do allow osmotrophy, of course, as sort of the basal characteristic of organisms in the model. This would be modeling that scenario where the stem group of the eukaryotic lineage has not developed phagotrophy yet. It hasn't developed feeding, but it's living by osmotrophy only. So that would be that in between the feca and the leca that Susan showed in her model. And if we do that, our model results with a variety of different mixing rates show, as you might expect, that as nutrient concentrations increase, nutrient concentrations as phosphate increasing up in this action, as nutrient concentrations increase, the size of the organisms increase. I'm sorry, I missed it there. Here, this is size of the organism. Down here, you can sort of see. You can sort of see them broadcast up here in this scale. Picoplankton, nanoplankton, microplankton, different mixing rates. But no matter what modeling scenario we choose, one thing I need to mention too. What I do is I highlight three different nutrient levels here. This nutrient level here is what we diagnose to be basically a modern type, modern concentration of phosphate. This would be 10 times less than modern phosphate, and this would be 100 times less than modern phosphate, sort of entertaining this idea that the phosphorus levels during the past, especially during the time of eukaryote evolution, might have been considerably less than today. Okay, so when we look at this, what we see is that in the absence of grazing or in the absence of eukaryotic phototrophy, we simply don't get organisms larger than the picoplankton. So we're dealing with organisms restricted to basically the prokaryotic size. So this would not seem to be compatible with the geologic record of large sizes that we find preserved in the Acrotart record. Okay, the next scenario would be We have picoplankton phototrophy. We allow grazing, but no algal photosynthesis. So that means no photosynthesis in the size range greater than picoplankton. And this would be consistent with the evolutionary scenario where we had a stem group that had, or maybe even just the beginning of LICA, where we had grazing possibility as a eukaryotic trait, but without photosynthesis. So algal photosynthesis hasn't yet evolved. And you can see that there's a spectrum of possible size ranges associated with elevated nutrient concentrations. Some of the mixing models give lower amounts of organisms in the high size range. But basically, this model would seem to be allowable based on the geologic record. We do see evidence in the modeling for organisms which can become large. So let's just keep that one in mind. And then we have the full model, which is with osmotrophy, algal photosynthesis, and grazing. And this model, of course, allows us a full size spectrum of organisms, especially at higher nutrient concentrations. So this model, too, would also seem to be consistent with the geologic record. So what we would like to do is now to try to partition our arguments between the idea that this sort of time where we have these large eukaryotic microfossils, this sort of Mesoproterozoic mainly time. We want to decide whether or not algal photosynthesis was a part of this ecosystem. Well, to do that, we go back to the geologic record, and I mentioned Joachim Brock's exquisite work on the extraction of bitumen from rocks of all ages and trying to figure out where sterines were present. We've done some of this work too, and when we look at rocks from the 1.4 million-year-old Jamaling Formation, if we try to extract bitumens from these rocks, we also don't find sterine biomarkers. But if we take the remains from after we extract the bitumen, it's called kerogen, we heat that up sequentially, we find that when we go up in temperature, here is the bitumen, there's no sterines, this is the sterine bunch down here. We go up in temperature. When we heat up this kerogen into about 440 degrees, in other words, a very, very high temperature pyrolysis, that we start to liberate sterines from the kerogen. So the sterines are there, they're just tightly bound to the kerogen, so you'd never see them in a bitumen extract. This work has been largely ignored by the geological community until further, because it certainly puts a wrench into our thinking about how eukaryotic ecosystems have evolved. But this is also very, very carefully done work, and I feel very, very confident that in this particular place, in this particular setting, that there are relatively large amounts of sterines encapsulated and captured strongly into the kerogens there. So I think that this is at least one indication that phototrophs were present in the system, and that's just to show you that this is where the Jamaling sterines lie, and this is the distribution of different sterine types. This is red. These are the red algae, green algae, and you can see that it looks like this would be consistent with a mix of red and green algae in the ecosystem at that time. And this isn't so crazy based on other evidence. This is from Stefan Benkman's work, where he finds what he interprets as red algae in even older rocks from the Vindian of India. And Andy Knoll has been associated with some work from similarly aged rocks from North China, which have also been interpreted as algae. So the idea that algae were in the ecosystem at that time seems to be compatible with at least some of the evidence that's emerging from the geologic record. So what that means is that the full ecosystem model might be the most appropriate for our interpretations of how the ecosystems might have functioned at that time. So now what we want to do is to just see what this model actually tells us with respect to the magnitude of eukaryotes in this ecosystem, this ancient ecosystem. So what we do is we have the high mixing model, the low mixing model. At different phosphorus levels, low phosphorus is 100 times less than today. Intermediate's 10 times less than today. High is today. And if we look at the distribution of biomass in different size classes, okay, we can find that some models will contain most of their biomass in the picoplankton. But most of the models actually have a rather, not even, but at least a distribution of biomass across what we would consider to be a range of prokaryotes into eukaryotic sized organisms. And when we get into the microplankton, that would be the size range where we actually see preserved microfossils. We also see that these models, especially at higher phosphorus levels, allow us a distribution into that range. So it appears that as long as we accept that these large organisms preserved in the geologic record, these large eukaryotic organisms can be interpreted in this way, that it seems their presence really demands that eukaryotes were rather significant players in the ecosystems during Mesoproterozoic times. And, well, how important was algal photosynthesis then? And this is I apologize, this is a little bit difficult to see through, but I'll help you to do that. What I've written here, or accumulated here, is the percentage of how carbon is acquired through the different size classes in the different models. So in other words, if we go to this model here, prokaryotic photosynthesis is responsible for almost 63% of the carbon accumulation into biomass. DOC uptake is about 26%. Phagocytosis is very small. And you can see that there isn't so much in these other size ranges. If we go up here to a high mixing scenario with high P, you can see that prokaryotic photosynthesis, or small size range photosynthesis, is equivalent for 5%. DOC uptake in the smaller size classes is responsible for a lot. But why don't we focus on the light, because we're talking about photosynthesis. We can see that the picoplankton, the nanoplankton, and the microplankton together are associated with sort of equal amounts of photosynthesis in this environment. And if we just sort of gauge through all of our different model results, focusing on the light, we can see that, at least from this modeling perspective, that eukaryotic-sized algae would have been responsible for a significant amount of the photosynthesis in these modeled environments, except under some very few modeling conditions. So with that, I come with some conclusions. I would say that late Paleoproterozoic and Mesoproterozoic ecosystems likely contain eukaryotic grazers and algae. That if this is true, then eukaryotes may have been important players in late Paleoproterozoic and Mesoproterozoic ecosystems. This is true to marine phosphate levels that down to about 100 times less than today. And what about this rise of algae? What about this finding of Joachim Brock's? Well, I would argue that maybe a preservational artifact where we've already heard today that some biomarkers become unstable over time. Well, sterines may also become unstable just over a longer period of time, which is why we find them tightly bound to kerogens in the Jameling Formation, but not associated with the bitumens. And I would like to thank a number of people associated with the modeling and the Velam Foundation for support, but I'd mainly like to congratulate you, Andy, on your fine day. Yep.